Be it a newborn baby, an adult, or an elderly person, our brains enable us to sense our environment, react to it, and is changed by experience, just like what I'm going through right now. This process is called plasticity and is an underlying mechanism in learning. When this process goes wrong, our brains can show abnormal plasticity, resulting in neurological disorders. One such disorder is Rett Syndrome, which is diagnosed as a neurodevelopmental disorder that affects 1 in 10,000 girls worldwide. Many girls and women with Rett Syndrome exhibit atypical speech, social communication, movement, and sensory perception issues. Mutations in the gene MECP2 causes Rett Syndrome. MECP2, which is present in the nucleus of the cells, regulates the expression of genes thought to be important for brain plasticity. The microscopy image here shows beautiful punctate expression of MECP2 in green in the nucleus of GABAergic cells in the brain. A major question is how mutations in MECP2 in the nucleus of the brain cells will result in neurological phenotypes, quite a diverse population of them. So we use mouse as a model system to study this. MECP2 is expressed in the nucleus of brain cells of mice as well. And when deleted, we'll have similar expression patterns. So we use a model of maternal plasticity, which is adult plasticity. As we are all aware, parents of many different species with fully grown brains and functional brains adapt and express plasticity to take care of their offspring. One test of this is maternal care in pup retrieval assay. When the pups are dispersed away from the nest, mice then exhibit maternal care where they will retrieve the pups back into the nest. In our assay, we use nanny mice to see if they will take care of the pups that are not their own, which is a test for plasticity in a new social setting. The human equivalent is shown here. My daughter here is acting as a pup, and my student here, Brett, who's not used to taking care of children, is trying to approach her with caution and concern, while my mother is well-versed in taking care of kids and responding to the situation with plasticity. When we look at this in control mice, nannies here, you can see that the adult pup goes to the pup over there, tries to pick it up, usually tries to find it by sound and smell, and then brings it over to the nest where it's already retrieved all the other pups. When we look at the MECP2 head pup, nannies in the next assay here, she's actually standing on top of a pup right there, and she will go and look for another pup over there, but she actually doesn't retrieve them into the nest. You can see all the pups are still scattered all over this cage. So what are the neural substrates that are different between these two different genotypes that allows for these differences in behavior? We previously found that there is a structure called perineuronal nets, which are labeled PNNs here, which are actually increased in expression in these het nanny mice. This is a microscopy image showing that abnormal increase in the het mice compared to the control mice. When we counted for these sections, by looking at multiple brain sections, we noticed that the het nanny mice had more of these just after the experience with the pups, not too far after or not before, suggesting that there was abnormal plasticity that's going on in the brains of these mice and not in the wild-type brains at all. So, since we had an increased expression, we wanted to look at PNNs here. What are they? They are extracellular matrix structures that are fence-like, and they can help form synapses, or they can actually inhibit formation of these connections. They're shown in green over here, and they're on GABAergic neurons, which are the red colored ones on the bottom slide down there. Since we had this increased expression of the PNNs in these brains, what we decided to do was to inject something called chondroitinase ABC, which is an enzyme, directly into the brains of these mice, which will reduce the expression of these perineural nets in these ones. And you can see that in the bottom slide, where the expression of the PNNs have actually reduced in these mice. When we did this, we could actually rescue or improve some of the behavior of these MECP2 heterozygous females in their pup retrieval behavior, shown in the red circles that you're seeing here. Each individual circle is a mice that had actually performed this particular behavior. So since this is a very complex behavior, we speculate that many other brain regions are important. So right now, we are actually doing serial sectioning and 3D reconstructions of all of these mouse brains to try to figure out what brain regions are affected in this process. We're seeing very specific hemisphere differences and region-specific differences in these mice currently. So our work suggests that PNNs act as key neural motifs that are substrates for plasticity in specific brain regions during learning and consolidations of these new behaviors. Without MECP2, this PNN expression is dysregulated in these processes in learning, resulting in stereotypic phenotypes. So the identification of these networks is not essential only for Rett syndrome, but also for other disorders like autism spectrum disorder, mainly because the pathology of symptoms such as language impairment, repetitive behaviors, and social deficits are also found in other disorders, not just in Rett syndrome or autism. With new genetic sequencing techniques, thousands of genes have actually been identified to cause neurodevelopmental disorders. Many of these genes have very different functions, as seen in the different colors shown in this diagram. So the next question would be, how do we try to wrangle all of these things together? And we think identifying neural circuitry motifs that are common across these genetic abnormalities is important. In summary, a major part of identifying these networks is going to be in looking at through the lens of brain development and to try to connect the genotypes to phenotype. And actually, we found Rett syndrome and neural net connections based on earlier development studies. I would like to thank all the undergraduate students, shown here in orange, for actually their curiosity and performance in doing most of these work. Find out more about Rett syndrome and how to support the girls and their families in East Tennessee through our website listed here. Thank you to the organizers for this opportunity and my family for modeling all of those pictures that we did. Thank you.